Cool Gyan, CBSE Class 12, Revision Notes, Class Chapter 10, Microbes in Human Welfare. Revision Notes for CBSE Class 12 Biology, Chapter 10. Microbes in household products, industrial products and sewage treatment. Microbes are the major components of biological systems on the earth. They are present everywhere, that is, in soil, water, air, inside our bodies and those of other animals and plants. They can also be found deep inside the geysers, thermal vents, deep in soil, under the layer of snow, and in highly acidic environments. Microbes are so minute that they cannot be seen by naked eyes. Various types of microbes are protozoa, bacteria, fungi, virus, viroids, and prions. Microbes in household products. The common products obtained by the use of microbes are curd, dough, toddy, cheese, etc. Curd is formed by adding bacteria such as lactobacillus and others, commonly called as lactic acid-based bacteria, in milk. A small amount of curd is added to the fresh milk as a starter, which contains millions of lactic acid bacteria. Lactic acid bacteria at suitable temperature multiply and convert milk into curd, which also improves nutritional quality by increasing vitamin B12. During growth, lactic acid bacteria that coagulates and partially digest the milk proteins. Lactic acid bacteria also checks disease causing microbes in the stomach. Dough is formed by fermentation caused by bacteria. Dough used to make bread is fermented using baker's yeast, Saccharomyces cerevisiae. Carbon dioxide released during the process of fermentation gives the fluffy appearance to dough. Dough is also used to make food like idli, dosa, in addition to different types of breads. Toddy is a traditional drink of southern India. It is made by fermentation of sap from palm trees by bacteria. A number of other drinks and food are also made by fermentation. Cheese is made by partial degradation of milk using different microbes. Swiss cheese is made by a bacterium, Propionibacterium stremanii. The large holes in these cheese are due to production of a large amount of carbon dioxide by the bacterium. Roquefort cheese is made by ripening with the fungi Penicillium roqueforti to obtain a specific flavor. Microbes are also used to ferment fish, soya bean, and bamboo shoots to make food. Microbes in industrial products have immense importance. The main industrial products obtained from microbes are fermented beverages, antibiotics, organic acids, alcohol, enzymes, and bioactive molecules, etc. Production of these products on industrial scale needs growing microbes in very large vessels called fermenters. Fermented beverages are wine, beer, whiskey, brandy, and rum. These are obtained by fermenting malted cereals and fruit juices with Saccharomyces cerevisiae or brewer yeast to produce ethanol. Variety of alcoholic drink depend on the type of raw materials used and the type of processing. Wine and beer are produced without distillation. Whiskey, brandy, and rum are produced by the distillation of the fermented broth. Antibiotics, antiagents, and biolife are the chemical substances which are produced by some microbes and can kill or retard the growth of other disease-causing microbes. Penicillin was the first antibiotic discovered by Alexander Fleming. Fleming discovered penicillin while working on Staphylococcus bacteria. He observed a mold growing in one of his unwashed culture plates around which staphylococci could not grow. He found out that it was due to a chemical produced by the mold. He named it penicillin after the mold Penicillium notatum. Ernest Chain and Howard Florey discovered full potential of this antibiotic. Penicillin was extensively used during World War II. Fleming, Chain and Florey were awarded Nobel Prize in 1945 for this discovery. Antibiotics are used to cure deadly diseases such as plague, whooping cough, diphtheria and leprosy. Organic acids are produced by the microbial metabolic action. Important ones are citric acid, Aspergillus niger fungi, acetic acid, Acetobacter aceti bacteria, butyric acid, Clostridium botulinum bacteria, lactic acid, Lactobacillus bacteria. Ethanol is produced by yeast, Saccharomyces cerevisiae, on commercial scale. Enzymes used in various fields are also produced by microbes as given below. Lipase used in detergent formulations and help in removing oily stains from the laundry. Pectinase and protease used for clarifying bottled juice. Streptokinase produced by Streptococcus and modified by genetic engineering is used as a clot buster for removing clot from blood vessels of patients who have undergone myocardial infarction leading to heart attack. Bioactive molecules produced by microbes are cyclosporine, produced by Trichoderma polysporum fungus. It is used as immunosuppressive agent in organ transplant patients. Statin produced by Monococcus purpureus yeast is used as a blood cholesterol lowering agent. It acts by competitively inhibiting the enzymes responsible for the synthesis of cholesterol. Microbes in sewage treatment play major role. Sewage is the municipal wastewater containing mainly human bacteria. It contains large quantity of organic matter and pathogenic microbes. Therefore, before discharging into natural bodies, sewage needs to be made less polluting. Sewage treatment is carried out in sewage treatment plants (STPs) in following steps. Primary treatment of sewages. This step involves physical removal of large and small particles from sewage through filtration and sedimentation. Floating debris is removed by sequential filtration by passing through wire mesh screens. After this, the grit, soil and small pebbles is removed by sedimentation in settling tanks. The sediment is called primary sludge and supernatant forms the primary effluent. The effluent is taken for secondary treatment. Secondary treatment. Biological treatment of sewage is done on primary effluent. Primary effluent is passed into large aeration tanks with constant mechanical agitation and air supply. This allows vigorous growth of useful aerobic microbes into flourish masses of bacteria associated with fungal filaments to form mesh-like structures. These microbes consume major part of organic matter in the effluent while growing. This reduces the biochemical oxygen demand (BOD) of the effluent. When BOD of sewage gets reduced, it is passed into settling tank. The bacterial flows settle in tank and the sediment is called activated sludge. A small amount of activated sludge is pumped back into the aeration tank to serve as inoculum. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digester. In sludge digesters, other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. During this process, bacteria produce a mixture of gases such as methane, hydrogen sulfide, and the carbon dioxide, which forms biogas, can be used as source of energy. The effluent from secondary treatments is generally released into natural water bodies. Biochemical oxygen demand refers to the amount of the oxygen that would be consumed if all the organic matter in 1 liter of water is oxidized by bacteria. BOD measures the rate of uptake of oxygen by the microbes in a sample of water. Indirectly, it measures the organic matter present in the water. More BOD of wastewater indicates more polluting potential. Due to the increasing urbanization, sewage is being produced in larger quantities, so the untreated sewage is often discharged directly into rivers, leading to their pollution and increase in waterborne diseases. The Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save the major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage can be discharged in the river.